Our laboratory is interested in a subset of lymphocytes called natural killer cells, or NK cells, which are involved in the recognition of tumor cells and in the elimination of viruses. We have identified a gene which is very important for the regulation of the natural killer cell functions. And in particular, when this gene is inactivated, natural killer cells are more functional, are more potent to eliminate tumor cells. In addition, animals which have this mutation are more resistant to viral infections.
So we use state-of-the-art genetics technology, in particular deep sequencing, to resequence entirely the genome of the mouse. And we found the gene which was responsible for this mutation. And this was the second surprise. The first surprise that we could manipulate the mouse so that it's actually better in fighting against microbial infection and cancer. The second surprise that we could actually identify a gene which was previously shown to be an activating gene for NK cells. But at the end of the day, when one deletes this gene, the result is that the NK cells are actually more powerful to fight against this infection. So this was the second surprise. NK cell biology can be explained in one of two ways. Actually, two different steps. The first step is development, maturation, and acquisition of NK cell effector function. And the second step is really effector function of NK cells by themselves. So our study shows that one can reprogram NK cells during their developmental phase. And by targeting a gene which, again, encodes for an activating receptor, the result of that is that NK cells are in fact boosted. We found that this hyperactivation of NK cells, this boost of NK cell function, has actually a cost.
So if natural killer cell response is too potent, it can affect the adaptive immune response, and in particular the T cell response.
So the intuitive thinking that says that it's good to boost the innate immune response is actually very intuitive and probably good in many instances. But what our studies are showing is that in fact this has a limit. And in particular in our case, when you have hyperactivated NK cells, then you don't have as much of adaptive immune response afterwards. So we have tried to recapitulate our genetic studies using drugs. And in this particular case, the drug was an antibody. And we are very pleased to see that one could recapitulate the entire phenotype using antibodies that block this activating receptor. So this opens new avenues for therapeutic applications, in particular in patients with immune deficiencies, acquired or like for HIV patients, for example, or maybe also open allogeneic graft.
The next step of our study will be to understand the molecular mechanisms behind this hyperreactivity, and also to find the clinical conditions in which we could use this strategy as a treatment.